If you chop the arm off a human being, it doesn't just spontaneously grow into a brand new, fully functioning person.
No, thankfully it does not.
Right. Biology just, doesn't work that way for us. Once our cells figure out they are part of an arm, they are completely locked into that identity. But if you take a pair of scissors and chop the leaf or like the stem off a Coleus plant and just drop it in a glass of water, it performs this wild biological miracle.
It really is a miracle. It literally reprograms its own tissue to grow new roots.
Yeah, completely cloning itself.
Exactly. And the scientific term for that is totipotency. Unlike most human cells, many plant cells actually retain the genetic ability to essentially reset themselves.
Just hit the reset button.
Right. They can generate an entirely new organism from just a single tiny fragment.
And people do this all the time. I mean, if you've ever stared at a glass jar on your kitchen counter, just waiting a month for a stubborn houseplant cutting to sprout a single pathetic little white root, you have participated in this cloning process.
Oh, we've all been there with the kitchen counter jar.
Oh, 100%. But today, we're looking at how one gardener managed to hack that biological process to work in just 4 days.
4 days. It's incredible.
It is. We are exploring a masterclass in everyday science, drawn from an excerpt of a blog post titled Hormonal Pre-Soaking Techniques for Accelerated Coleus Propagation.
Yeah, and this comes from a highly detailed series called Notes on Gardening. We are specifically analyzing Part 889.
Part 889, that's just wild.
It is a lot. It was published today, actually, May 9, 2026, by an author known simply as Ang.
The volume of that documentation alone is crazy. I mean, Part 889 implies a level of dedication you usually only see in, professional laboratory settings.
Oh, absolutely.
So our mission for this deep dive is to unpack how a surprisingly simple tweak in the normal propagation routine led to these drastically accelerated results. And more importantly, we're going to break down how the author used rigorous scientific variable control to prove the biological mechanics behind why this tweak worked so well.
Which is the best part, honestly.
It really is. Okay, let's unpack this. Ang is actually following up on a previous post, Part 884, where they started detailing a modified method for propagating these Coleus plants.
Right, because the standard protocol for water propagation is familiar to almost anyone who keeps houseplants. You take your cutting, you put it in a vessel of water, and, well, you wait.
You wait forever.
Right. But what Ang is doing here fundamentally challenges that assumed timeline. They're inserting a highly concentrated intervention before the cutting ever sees its final growing jar.
And the core method they are testing involves taking the fresh plant cuttings and soaking them in a liquid rooting hormone for a full 24 hours at step 1. But I want to pause here because we throw the phrase rooting hormone around like it's a magic spell.
We do. We really do.
I understand the experiment setup, but what is that chemical actually doing to the plant on a cellular level through those 24 hours?
That is the crucial question. So when we talk about rooting hormones, we are typically talking about a class of plant hormones called auxins.
Auxins.
Yeah. And the most common synthetic version used by gardeners is indole-3 butyric acid, or IBA. To use a technology metaphor, you can think of an auxin as a highly aggressive software update for the plant's cells.
Oh, wow. So like a software update that overwrites the plant's current programming.
That is a perfect way to visualize it. I mean, when you cut a stem, the cells at the wound site are exposed. The plant's immediate biological priority is usually just to heal that wound to survive.
Just patch the hole.
Exactly. But when you submerge that wound in a concentrated solution of synthetic auxins, those chemicals penetrate the tissue. They target the undifferentiated stem cells, the meristematic tissue.
Okay.
And the auxin floods the cellular receptors and essentially overrides the plant's default programming, forcing those cells to differentiate into root tissue.
So by doing a dedicated 24-hour liquid pre-soak, Ang isn't just giving the plant a gentle suggestion to grow roots. They are locking the plant in a room and blasting that grow root software update into its system at maximum volume before the traditional water propagation phase even begins.
Spot on, because a standard method is to just dip the cutting into a hormone powder and immediately plant it.
Right, just a quick dip.
Yeah, but a 24-hour liquid saturation ensures the vascular system of the cutting has fully drawn the auxins up into its stem. The biological signal is inescapable.
Man, inescapable root growth. This brings us to a specific decision Ang makes in the setup of this experiment, which might honestly be my favorite part of the entire text.
Oh, the omission.
Yes. It's not about what they added to the process. It's about what they explicitly chose to leave out. The source mentions a deliberate omission. Ang writes, I decided against using an air stone to oxygenate the water, for now to focus on selected factors first.
That single sentence demonstrates a profound grasp of scientific restraint.
I am so glad you agree, because this is where a lot of casual experiments completely fall apart. I like to think of this like a chef trying to invent a new marinade for a steak.
Okay, I like where this is going.
Right. So if that chef wants to know exactly what makes the steak taste incredible, they can't throw 10 different bold spices into the mixing bowl all at once. If they add minced garlic, crushed onion, cumin, smoked paprika, and like a huge handful of brown sugar, and the steak comes off the grill tasting like a masterpiece, the chef actually has a massive problem.
Because they have no idea what actually works.
Exactly.
Yeah.
They have no idea which ingredient did the heavy lifting. Was the sugar the secret? Was it the cumin? The data is entirely muddy.
The chef analogy works beautifully for explaining variable isolation, because the natural human temptation, especially when you are emotionally invested in a successful outcome, is to throw every single advantage at the problem simultaneously.
Oh, for sure. You just want the plant to live.
Right. A gardener wants their plant to survive, so the instinct is to use the heavy hormone soak, deploy the air stone, provide the most expensive fertilizer, and put it under a fancy grow light.
But if you do all of that and the roots explode with growth, you've learned absolutely nothing.
Nothing at all.
You've created a successful recipe, but you have zero understanding of the underlying mechanics. Let's actually talk about that air stone, though. Why would someone use an air stone in a jar of water in the first place?
Well, it comes down to cellular respiration. We tend to forget that roots need to breathe.
Even in water.
Yes, even though they are submerged in water, root cells require oxygen to convert the sugars produced by the plant's leaves into usable energy. In a stagnant jar of water, the dissolved oxygen is depleted very quickly.
Which leads to rot, right?
Exactly. When the oxygen is gone, anaerobic bacteria begin to thrive, which is precisely what causes cuttings to turn to black mush and rot. An air stone, like you would use in a fish aquarium, constantly agitates the water, increasing the surface area at the top of the jar and allowing fresh oxygen to dissolve into the liquid.
So an air stone is highly beneficial.
Highly beneficial.
Wow, okay. So by intentionally removing a tool that is known to promote healthy root environments, Ang is guaranteeing that the resulting data won't be compromised. They are holding back on adding extra environmental advantages so that any changes in the plant's behavior can be directly traced back to the specific variables they are actually testing.
Precisely. And the text explicitly outlines the variables chosen for the test. Instead of playing with oxygenation, Ang experimented by placing the propagation jars into two distinctly different microclimates and utilizing various jar sizes.
To see if water volume or light intensity would alter the hormone's effectiveness.
Exactly.
So they set up a baseline intervention, the 24-hour Oxen software update, and then they test how the physical environment interacts with that biological override. They put some jars in higher indirect light and some in a more shaded area.
Effectively building a control scenario right there on their windowsill.
The experimental design is so elegant.
It really is elegant. You establish constant, which is the pre-soak, and then you introduce environmental friction. The goal is to observe whether the environment dictates the outcome or if the biological constant overpowers the environment.
And the timeline of the results from this elegant design is what really shocked me. According to the text, after only 6 days, the visual evidence is overwhelming. Ang notes that the new root systems are sprouting fast, bushy, and strong.
Those three adjectives describe a biological gold standard for propagation.
Really, a gold standard.
Oh, absolutely. When you attempt to root cuttings in plain water, the initial root structures are often highly delicate. They tend to be spindly, translucent, and incredibly fragile.
Which makes transplanting them terrifying.
Exactly. It creates a huge risk of shock when you eventually try to transplant them into dense soil.
So when Ang describes them as bushy, what does that actually indicate about the plant's health? Is it just an aesthetic observation, or is there a real functional advantage there?
The bushiness implies the rapid development of lateral roots and root hairs, and this is functionally massive. A single long taproot has a very limited surface area.
Okay, that makes sense.
But bushy, branching root systems exponentially increase the total surface area available for the plant to absorb water and essential nutrients. Fast validates the accelerated time frame. Bushy confirms optimal nutrient absorption capability, and strong implies cellular density and resilience.
So this technique isn't just about shaving days off the calendar.
Not at all. It is engineering a biologically superior foundation for the plant's future life.
It's not just a speed hack, it's a quality multiplier. And Ang initially observes that it seems like these tweaked environmental factors played a role. They call out the hormone pre-soak, the higher indirect light, and the larger jars as the combination inducing this massive growth.
Right, the initial visual check confirms that the plants in the optimal conditions, the higher indirect light and the larger water volume, are exploding with cellular activity after the soak.
Well, here's where it gets really interesting. Because Ang didn't just coddle the cuttings in those ideal conditions. We have to look at the jars that were left in the entirely different microclimate.
The shaded area.
Yes, the shaded area. The text reveals something completely counterintuitive. The roots in the shaded jars sprouted just as vigorously as the ones sitting in the optimal, higher, indirect light.
And this specific observation is the pivot point of the entire process. This is the exact moment where passive observation transforms into active scientific deduction.
Let me push back on this for a second, though, because a skeptic might look at this and say the entire experiment is flawed.
Okay, let's hear the skeptic's view.
If the amount of light didn't matter and the size of the jar didn't matter, couldn't the simple answer be that Coleus is just a freakishly fast-growing weed?
That's a fair question.
Like maybe this specific species of plant is biologically hardwired to push out massive roots in 6 days, regardless of what kind of water you put it in or whether you soak it at all.
It's a great point, but the author's own meticulous documentation preemptively dismantles that skepticism. Remember the overarching context. This is part 889 of a continuous journal. Ang explicitly references part 884, which detailed their transition to including this specific soak. That historical data serves as an embedded control group.
I see the logic now. They already have the baseline data for how Coleus behaves under normal conditions.
Exactly. Ang knows intimately how long an untreated Coleus cutting takes to root on that specific windowsill. The explosive 6-day growth timeline is categorized as a new phenomenon within the context of their own garden's historical record.
So they look at the jars in the bright, indirect light, and they look at the jars in the shade, and they observe the exact same fast, bushy, and strong biological response in both environments.
Both of them.
The environmental factors were vastly different, but the aggressive growth remained entirely constant.
And the deduction is inescapable at that point. What's fascinating here is, because the lighting and the jar size varied without altering the outcome, those environmental variables cannot be the primary catalyst for the accelerated cellular division.
The environment is effectively a red herring.
Precisely.
Ang deduces that the universal constant connecting all the jars is the true engine driving the result. And that constant is the 24-hour auxin pre-soak paired with a fascinating secondary detail: the act of reusing that diluted hormone water to fill the propagation jars.
And that secondary detail shifts our understanding of how the hormone functions in this specific setup. By transferring the cutting along with the water it just soaked in, the plant isn't merely receiving a single temporary jolt of chemical instructions.
Right. The text actually says we can deduce that the 24-hour pre-soak and reusing that diluted hormone water is the real factor here. I am fascinated by the mechanics of that. Does reusing the water change how the plant processes the hormone?
It creates a sustained chemical environment. We talked about the 24-hour soak being an aggressive software update.
Right.
The initial concentrated immersion shocks the stem cells into differentiating. But by placing the cutting into a jar filled with that same diluted water, the plant is continuously bathed in a maintenance dose of the auxin.
A maintenance dose.
Exactly. It ensures the biological signal telling the plant to prioritize root growth never diminishes while the new tissues are forming.
Man, it isolates a highly specific biological trigger, while simultaneously disproving the long-held assumption that lighting and container volume were the most important factors for early root development. Ang writes, honestly, I'm quite impressed by how much of a difference a simple soak makes.
You can practically hear the satisfaction of the data clearing up the muddy waters.
You really can. The result completely demystifies the propagation process. It replaces the traditional reliance on luck and perfect windowsill real estate with a repeatable, verifiable biological technique.
It's science in action.
So what does this all mean? We have a rigorously controlled experiment, a clear deduction isolating the cause of the accelerated growth, and a biologically superior root system. But why does this specific finding matter for you, the listener? And what are the implications for the future of how we handle plant propagation?
The true value lies in the scalability of the concept. Near the conclusion of the post, Ang shifts the focus from the specific behavior of the Coleus plant to a broader botanical application. They state, this isn't just for Coleus, it can likely be applied to any cuttings you propagate in water.
Proposing a universal hack for water propagation is a massive takeaway.
It really is.
If you are trying to clone a pothos vine, a rare monstera, or even just trying to root a sprig of basil you brought home from the grocery store, Ang is suggesting that this 24-hour auxin saturation is a foundational shift in methodology.
And the most practical implication of that methodological shift is the complete restructuring of time. We are talking about fundamentally altering the expected timeline of a natural biological process.
Which is mind-blowing.
Based on these findings, Ang notes that these rooted cuttings no longer have to sit in a fragile aquatic state for weeks on end. They are ready for soil. They even hypothesized that the timeline could be compressed further, suggesting the cuttings could have been safely potted in just 4 to 5 days.
Shrinking a month-long waiting game into 4 or 5 days is wild efficiency.
Yeah.
You could theoretically prep a cutting on a Friday evening, apply the hormone soak, and have a biologically robust, soil-ready plant to pot by the following Wednesday.
The efficiency is undeniable. However, the most compelling aspect of this entire source text isn't necessarily the 4-day timeline itself.
Oh, what is it?
The true highlight is Ang's cognitive reaction to their own success.
Ah, right. They achieve a massive reduction in the timeline, but they completely refuse to just declare victory, write down the new rule, and walk away.
If we connect this to the bigger picture, Ang embodies the relentless, iterative curiosity that defines a true learner. They have just executed what they describe as a significant success and insightful experiment.
And most people would stop right there?
The vast majority of people would stop testing at that exact moment. They would permanently adopt the 24-hour soak as their new unquestionable law of gardening. Ang, instead, immediately begins hunting for the next bottleneck to eliminate.
The final notes of the text point straight to the next frontier. They write, soaking your cuttings in rooting hormone for 24 hours, or even as little as 2 hours, we'll need to experiment that.
I love that part.
They literally just finished optimizing the process, and their immediate instinct is to optimize the optimization.
It is a brilliant display of the scientific mindset. They proved beyond a doubt that the 24-hour baseline works perfectly. Now, they are putting their own newly established rule under the microscope. Is a full 24 hours actually a biological necessity?
Right, because we don't actually know the maximum cellular absorption rate of the auxin in this specific setup. What if the meristematic tissue in the plant stem reaches total chemical saturation in just like 120 minutes?
If the biological limit is reached in 2 hours, then the remaining 22 hours of the soak are functionally redundant.
Wow.
By questioning the necessity of their own baseline, Ang is preparing to run the exact same rigorous, variable-controlled framework all over again, solely to see if they can claw back another 22 hours of sheer efficiency.
It perfectly illustrates that the scientific method is not a physical location or a multi-million-dollar laboratory. It is a way of thinking. You solve one specific problem, and the mechanics of that solution immediately present a more refined question to investigate.
Exactly.
Ang is applying the exact same logical, rigor-isolating variables, analyzing mechanisms, and optimizing outcomes to a glass jar of water that a pharmaceutical researcher applies to synthesizing a new compound.
The stakes of the experiment are vastly different, of course, but the cognitive architecture required to run it is identical. We watched a hypothesis get systematically proven and then immediately evolve into a sharper hypothesis.
Let's bring all of these concepts together. We started this deep dive looking at what seemed to be a mundane blog post about multiplying houseplants, but what we actually unpacked was a masterclass in challenging biological defaults.
We really did.
We moved from understanding how stem cells can be forcefully reprogrammed by an auxin soak to utilizing the chef's marinade strategy of variable control by eliminating the air stone. We watched as the data proved that environmental factors like light and jar size were irrelevant compared to the sustained chemical environment of the diluted hormone water.
Ultimately shrinking a month-long process into a hyper-accelerated 4-day timeline.
It serves as a powerful reminder that observation and controlled variables can completely reinvent the things we do on autopilot. If you have ever accepted a slow, frustrating process just because that's how it has always been done, this experiment proves you don't have to accept the default timeline for anything, provided you are willing to rigorously test the underlying mechanics.
You know, this raises an important question.
I like where this is going.
Ang realized that the standard 30-day waiting period for roots was simply an assumption. It was an accepted reality that completely shattered the moment the correct chemical pre-soak intervention was applied.
Right.
What other assumed timelines in our own lives operate under that same illusion?
That is a fascinating lens to apply to our own behavior.
What other traditionally slow processes might be drastically shortened if we analyze the mechanics? Whether it is learning a complex new technical skill for your career, pushing through a massive personal project, or trying to encode a difficult new daily habit into your routine.
We just accept that they take forever.
We frequently assume these developmental phases simply require months or years of grinding effort because conventional wisdom tells us they do. But what if they don't? What if there is a cognitive or structural catalyst we simply haven't isolated and tested yet?
Yeah.
What bottleneck in your own life might be drastically shattered if you could just discover the equivalent of a 24-hour pre-soak?
That is an incredible thought to leave on. The tools for profound discovery aren't locked away behind the sterile doors of a high-tech facility. Sometimes those revolutionary breakthroughs in how we approach growth happen when we simply decide to look really, really closely at a glass jar sitting quietly on a windowsill.